about the mechanics of flight, and really it's a question that I've been working on ever since I can remember, ever since I have entered academia. And it's a question that continues to yield just rich insights into insects, which of course this is the world of insects. We are a mere blip in their evolutionary history. So what I'm going to do is start by showing you a video. I like to show videos because once you see insects, then you begin to appreciate really what gets me so excited about it. And the video I'm going to show you contains just about everything, maybe not everything, but a lot of why people like me get so excited about this and why we see a career's worth of work to be done here, many careers worth of work. But what I'm going to show you is very mundane, something that you've seen all the time. There's a housefly, we just saw some outside, which will come and land on this. Now, getting a video like this is not easy, quite in the way that we see this all the time, but to capture it, to make sure that you have a couple of cameras focused exactly in the same place at exactly the time that you can get this fly to come and land, and we have tricks to do that, is what is difficult. And so this video was taken by a student intern, Sanjay before, when he was in the lab. He's now in University of Washington. So this is a housefly. I'll give you some quick facts. It flaps its wings 200 to 250 times a second. We're filming this at about 2,000 frames per second with a high-speed camera. And this is what it does. So here it comes, does this roll, and then lands. Okay, I'll show you this video one more time. What's interesting about this is that all of this action is happening in a matter of maybe 15, 20 wing strokes, so well within a blink of an eye. But you can appreciate how precise it is. And for it to be this precise, the fly has to do many things. And that's really what drives us. So there's a lot happening in terms of what the flies are doing with their wings. So wings are moving. As they move, they generate aerodynamic forces. They're controlling the torques generated around their body. But more importantly, maybe not more importantly, equally importantly, they are paying attention to their sensory world. And so that square that you saw is expanding on their retina, okay? And that's how the fly knows when it is close. And we've actually gone quite deep into this with Sujay about how flies land, but I won't talk about that today. Here's another video, and this gives you a sense. I've slowed it down much more, just to give you a sense of all the wonderful things that the fly does with its wings. So just watch this video carefully. Watch the wings. Simple video, just taking off, but there's tremendous amount of modulation of wing motion that's going on, okay, as the fly rights itself and balances. And so this is a really fine balancing act. Now, how does a fly manage to do this? One of the things, of course, is that it's paying attention to its visual environment. But the second very important aspect of this is that it is getting mechanosensory stimuli. So just as we have our inner ear system, it would be impossible for me to walk across the stage without my inner ear system, my vestibular system working properly. Flies too have a similar system. It's very different in its mechanism, but it serves the same purpose. Except, let's again remind ourselves that this animal must operate within milliseconds. Okay, we can afford to be much more sluggish. So this is the challenge really. And so what really gets us excited about insects and insect flight is the fact that A, it's really fast, and B, it's extremely precise. And usually we when we think about things, you think that if you increase speed, you lose precision, but somehow that's not happening here. I mean, I think it's an easy case to make that flight enabled the vast diversity that we see of insects. That flight was the key innovation that made insects. So if this is the sort of the life history of all animals, so to speak. So it's a phylogeny, but everything on the left here is insects. Okay, insects are vastly diverse. They are more than 60% of all multicellular animals, and this is just of what is known. Of course, so much is not known. I mean, I think we know less about the insects out there than we know about, say, mammals or other animals, birds, most of which have been documented. But insects continue to remain largely undocumented. So there's an estimated about 6 to 10 million species, of which more than 1 million are described, of which 60% are insects. What's also really interesting is that insects range vastly in size. Okay, and this is a question that I'm getting more and more interested in. I'll touch a little bit upon it. They also occupy vast variety of ecological niches. I think there's no part of the world where you won't find insects, and they are also very old. They've been around for at least 400 million years, and I suspect they'll be around longer than us. So just to focus a little bit on the size aspect. So this is the largest insect that has ever been found. It's a meganeurid dragonfly, and it has a wingspan, which means from one tip of the wing to the tip of the other wing, of about 65 centimeters. This is extraordinary. I joke that this thing could carry us if it were around. This was about 300 million years ago, during the Carboniferous period. It's thought that there was a oxygen blip, a lot of oxygen in the environment at the time, and that's what allowed this kind of large sizes to evolve. The largest extant insect today is the Queen Alexandra's birdwing, which has a wingspan of about 30 centimeters. And we have the southern birdwing in these parts of the world, which is not that different from this. So this is a pretty large. You can, if you just look at it from the corner of your eye, you might think it's a bird, a medium-sized bird. So that's large insects. Okay. On the other end of the spectrum is this wonderful insect. It's called the Megaphragma maripene. And just to give you a sense of scale, so this is 200 microns. So this is how big the insect is, and that's a cell, a single paramecium cell. And so you have here a multicellular animal that has antennae, it has eyes, it has wings. These wings are feathery, but at these scales, a feather is just like a paddle, and it is able to fly. And in fact, we are beginning to study not Megaphragma, but insects of this size, because it's just fascinating. So when we think of the question of insect flight, there's so many things to do. And how do you go about in a laboratory trying to study this? So you have, behavior is what drives us. So at the end of the day, this is what we are focused on. And we look at behavior in many different contexts. So insects are out there looking for mates, looking for food, they're guarding territories, they're navigating either in short distance or long distance. I think all of you are aware of long distance migrations that insects are capable of. And so it's in these contexts that you see insects, and then once you see something interesting, you want to bring it into the lab. And so that's essentially what we do. So the movie that you saw, the first movie that you saw, is an attempt at bringing a interesting behavior we see outside into the lab so we can study it. Now once we have reconstituted the behavior, then the questions that we tend to ask, at least in my lab, have to do with what are the sensory modalities that the insect is paying attention to. And it's not just one modality, but multiple modalities. They see, they sense, mechanical sense, they smell. All of these are being integrated by the central nervous system. There's a fair bit of work going on in the lab at this end. And then the central nervous system processes this information and generates a concert of muscle activity. And it's a very finely coordinated, conducted concert of muscle activity. And I'll touch upon that a little bit because it's a really important part of the mechanics. Now, the muscles are going to contract because they're listening to these motor neurons. And these muscles will then, because insects have an exoskeleton, unlike us, we have an endoskeleton, they have an exoskeleton. And so what they do is they pull on the exoskeletal parts. And that's all of this stuff here, where they will then move the thorax, and then that motion is somehow through a really interesting wing hinge transmitted into wing motion, transduced into wing motion. And this concert is absolutely fascinating, and we know very little about it. So I'm going to talk a little bit about that as well. And then, of course, the wings move, and they move in very subtle ways, as you've just seen. So the wing kinematics will then push the external fluid medium around, which will cause locomotor forces, which is what we see as the behavior. So that's really the full sort of extent of this. Now, there's many aspects of this when I talk about mechanics specifically. And of course, the point I want to make is mechanics is just one part of the question. I mean, all of this is going on, and we can't really ignore any of it. But if one is just talking about mechanics, then one is talking really about this whole part. So I spent a lot of my early years working on a question, almost obsessed with this question of how insects generate aerodynamic forces. So back when I was still a physicist, I was interested in these kinds of questions. And so what we did when I was a graduate student, this was in Mike Dickinson's lab in UC Berkeley, what we did was we constructed a robot. And what this was a different way of looking at the questions. Up until then, people used to study these kind of questions in a wind tunnel where they had a wing that was held steady. But we argued that that's not really recreating the conditions of flapping. And as you might have heard, there was an assertion made that, given the contemporary laws of aerodynamics as we understood them, we knew enough to fly planes, but we couldn't explain how bumblebees flew, or any insects flew for that matter. And there was some truth to that. And so we asked, what can we add to this if we actually bring in the flapping system? So this was just our way of trying to make sure that we brought flapping into the sort of experimental system. And so this was the wing. So this is a wing that's about 25 centimeters long. At the base of it, we attached force sensors, strain gauges, in 2 dimensions, so parallel and perpendicular to the wing. And then we had 3 motors that actuated this wing in 3 dimensions. And so this is what it looked like. Sorry. So this is, and the whole thing was dunked in a tank of mineral oil just to make sure that the Reynolds numbers were conserved. This is just a ratio of inertial to viscous forces. So this is what it looked like. So we could sense the forces. So we had the whole force record while it does that. And we also could fill the tank with bubbles. And this is better seen in this new version of RoboFly that we have now reconstituted in Purdue. Now it has a body and so on. But what you can see is these bubbles. And these bubbles can be tracked by a specific method called particle image velocimetry. And that's particle image velocity. So you can throw a laser sheet across, and then what you get is a picture of static bubbles. And if you take these pictures closely enough, then you can ask how the bubble configurations change. And it's really a method that requires computation. And so its time had come. By the time I was a grad student, we could do these kind of things. And so what you then get are these vector fields, velocity fields. Each point here is a vector. and you can already begin to see structures like this leading edge vortex here, and then if you take a derivative, a curl of this, then you get vorticity. So I won't talk about this, except to say that so we could see the flows, we could see the forces, and this allowed us in many ways to then understand how flapping was different from fixed wing. And we were able to then resolve the issue of how insects generated sufficient aerodynamic forces to fly. But this is old stuff. It's important because I'm going to take one result from here and talk about it in the context of miniaturization. Okay, so one of the results is that if you I won't go too much into the equations, but if you look at the equations, what happens is that if an insect wants to generate greater force, then it can do it in two ways. Either it can increase its amplitude, which is the excursion of the wing, or it can increase its frequency. Okay, and as it turns out, as you miniaturize the insect, it has to do both. But there's a point beyond which you cannot increase your amplitude, simply because the wings crash into each other. And so the only way it can now increase its flight forces, and it has to increase it relative to its body size because for various reasons. I won't go into that. So it has no choice but to increase its wing beat frequency. And in fact, if you look at the data on this, so you can think of mass as being a proxy for size or wing length as being a proxy for size, but in both cases, what you see is that as size decreases, wing beat frequency increases. And you can see that at its peak, it goes almost to 1000 hertz. Midges fly, for instance, at 1000 hertz. That's extraordinary, a wing beat every millisecond. So how do they do that? How can a nervous system, which is much more sluggish than this, be able to generate something that is of this extraordinary speed? Now, there's other interesting things about miniaturization, because not only is their ability to generate forces compromised, but also their ability to sense the world or to generate muscle commands. Energy loss is always a major problem. The smaller you are, the greater your surface area to volume ratio. And of course, water losses. There's such little water in them that they are always struggling against. And most of the times, miniature insects die because of dehydration. So these are just a few of the many challenges that insects deal with. So if you think about one very important sensory organ, antennae, which is something we work with, there are fewer segments, fewer sensillae. Odor perception is diffusion limited in this case. Their eyes have fewer units. As you know, insects have compound eyes, so there are fewer units, poorer resolution, smaller diameter lenses, and so they are diffraction limited. And so the body size, there's a simple eye called ocelli. I won't go into that, but that is somehow not affected. Wings are often reduced or become fringed, but remember that if you were to think about miniature insects, for it flapping in air is almost like us trying to move our hand through a jar of honey. And at that point, if it's something that viscous, then a fringe is as good as a paddle, as I mentioned. So that's something, but they have to, of course, enhance the wing beat frequency and so on. And then, there's other challenges at the more ecological level. How do they disperse? How do they migrate? How do they move about? And then thorax and flight muscles are also extremely under pressure to cope with loss of size or with miniaturization of size. And yet, these insects are really fantastic. I'm going to show you a video just to give you an appreciation of how much like ordinary insects they are. This is a wasp, parasitoid wasp, which is on the egg of another insect. Okay, so you can imagine how small it is. and you can see that it's exploring the egg, okay, with its antennae, and at some point it stops and then injects its eggs inside. Okay, so it's sensing, it's processing, it is doing everything that you would expect from an insect. This is just a close-up of the eye, and you can see that the eyes begin to lose the number of units because they simply cannot, each unit cannot become smaller than a certain degree because it's diffraction limited, and so they just compromise by losing units. So it's a very ill-resolved eye. Each of this unit you can think of as a pixel. And if you think of how they fly, so these videos are very hard to get, but you can see that it claps, it's just running out of space there. So it has to increase frequency. Okay, so how do they do this? How do they increase the frequency? If the nervous system can't act faster than say 2 or 3 milliseconds a single action potential, how are they able to increase the wing beat frequencies? And so this is a question that was resolved quite a while ago by Pringle above all. And what happens there is there are two types of muscles. So there are the larger insects like locusts, which have something called a synchronous muscle. And by synchronous, what we mean is for every nervous impulse, the muscle contracts once. And then there is beetles and bees, Hymenoptera, and flies, which have this thing called the asynchronous muscle, in which for every neural signal here, you have multiple wing beats. Okay? And so what you have is a resonant system, and I'm going to tell you how this resonant system is constructed. So first of all, what we need to know is that these muscles, the muscles in these specific insects which have been able to miniaturize, have an interesting property called delayed stretch activation. What that means is that when you take a muscle and you contract it, okay, it takes time to get activated. So if you take the muscle now and extend it, then after a certain delay, it's going to contract, it's going to generate force. Okay? And this is something that's seen, you can see this delay here as compared to here. This is the locust, this is beetles. This means that if you take now an insect, right, and so this is the thorax of the insect, and you have these two antagonistic set of muscles, and you pull its thorax in one direction, okay, either because, so if these muscles get a nervous impulse, then they contract, and now the thorax extends in this direction, right? And because it's extended, after some delay it's going to contract, and then this will extend, and then this will contract, and then this will extend, and this happens many times, okay? And that's what constitutes the resonance, okay? And so this is how we get the contraction of these dorsal longitudinal muscles and the dorsoventral muscles. These are called the power muscles. And when the DVMs, the dorsoventral muscles contract, the wing goes up. When the dorsal longitudinal muscles contract, the wings go down. And so you put these two together, you have a resonant box. Now you don't need as many neural impulses. Okay? Now I would argue that the combination of the myogenic muscle, the muscle in which a single neural impulse is able to generate many oscillations, and the indirect flight muscle architecture, where muscles are not directly connected to the wings but to the thorax, is what has allowed us to go what has allowed hyperdiversity. So if you look at all the hyperdiverse orders, the orders that have really been successful, if diversity is a measure of success, then all of these have this myogenic muscle and IFM architecture. But how are they fast and precise at the same time? And this was a question that I had this extraordinary student, Tanvi Devra, who joined my lab many years ago now, and she was the one who first posed this question: how can they be both fast and precise at the same time? There must be something going on. Can't be the nervous system. That can't be the whole answer. I mean, initially she thought that was the answer. She wanted to be a neurobiologist. But we focused on this one system. So this is a soldier fly, And I want you to see this particular structure here. It's called the haltear. This is the gyroscope. I told you about the vestibular system of insects. That's the vestibular system of the insects. And it's an organ. It's the hind wing that has been reduced to a mechanosensory organ. And what it does is it vibrates in a plane. Okay, when it vibrates and the insect turns, it experiences these forces called Coriolis forces. And that's how the insect knows how fast it's turning. Okay, I'll show that again. Notice that it is exactly beating out of phase with the wings. Okay, so they need to be really precise. Otherwise, this insect is going to start going around. You know, it will get vertigo. And so, if you look at the base of this haltear, focus closely, then you see these individual structures. Each of this is a sensory organ. Okay, it's called a campaniform sensilla. And fields of campaniform sensilla are experiencing strains due to Coriolis forces, which then are transmitted to the brain, and the brain interprets this as an angular turn. And so, these control muscles, there are muscles at the base of haltears which are being controlled by visual feedback, but this input, the input from the haltears goes into the wing motor neurons, or wing muscles then will respond to what the haltear is saying. And so, if you knock out the haltear, I need to speed up a bit, then this is what happens. Okay, so this is an insect that's simply unable to control its flight. Okay, whereas a normal insect is very nicely balanced. And if you just knock out the left haltear, then I'll just show you the left one, not the right one, but you'll see that it tumbles in one direction. If you knock out the right one, it tumbles in the other one. Okay. So, haltears are very important, and what Tanvi observed was they were beating exactly out of phase. So, the left and right wing are exactly in phase, but the haltear and the wing are exactly out of phase. And so, she argued that this must be, because she was a neurobiologist wannabe, she said, oh, it must be that the wing sensory neurons are driving the haltear motor neurons, or the haltear motor neurons are driving the wing motor neurons, or haltear sensory neurons are driving the wing motor neurons, or some neuron is telling the wing motor neuron to go down while the haltear to go up. But for completeness, she said that, well, it may be that they are just mechanically coupled. Okay. And so, herein comes the interesting observation. So, what she did then was ask, the last hypothesis is easiest to address. So, she said, what if I just look at a dead insect? And so, here is a dead insect, and what she does is moves their wings, such a simple experiment, but such a profound one. And you can see that the haltears move exactly out of phase. Now, if the insect thorax has this structure called scutellum, which connects the two bases, as you can see here. And so, she began to play around with this, and what I am going to show you is a movie that summarizes everything that we found. So,
allow me biomechanics of wing and haltear coordination in flies. Our study shows that precise coordination between wings and haltear is achieved not by the nervous system, but by mechanical connections within the thorax. The structure on the thorax, the scutellum, links the two wings. Rapid coordination of the indirect asynchronous flight muscles drive the motion of the scutellum, which causes simultaneous in-phase movement of both wings. When this link is severed, the two wings become uncoordinated, as shown in the next video. Thus, the scutellar linkage is necessary for wing-wing coordination. Although the two wings are not coordinated anymore, the haltears on each side oscillate antiphase to the ipsilateral wing. When the two wings become uncoordinated, the haltears also become mutually uncoordinated. We next showed that the wing-haltear coordination is mediated by a separate mechanical linkage, the subephemeral ridge, which connects the wing base to the haltear base. When the subephemeral ridge is intact, the wings and hot-ears oscillate exactly anti-phase with respect to each other. However, when this link is lesioned, wings and hot-ears become uncoordinated. Notice that the hot-ear continues to move through its full amplitude, driven by the asynchronous hot-ear muscles. The hot-ear on the right side, whose link remains intact as an internal control, continues to oscillate exactly antiphase with the contralateral wing. Thus, the subephemeral linkage between the wing and the hot-ear system on both sides are independent of each other. If the wings and hot-ears are constrained to move in synchrony by mechanical linkages, how do insects achieve control of just one wing at a time? To address this question, we propose the hypothesis that there exists a clutch at the base of each wing, which can engage and disengage the wing from the mechanical linkages. When the clutch is engaged on both sides, the two wings flap together. However, when the clutch is disengaged on one side, one wing remains folded, whereas the other can flap. Apart from the clutch, the base of the wing contains a gearbox. Once the wing is engaged, the gearbox controls the amplitude of each wing. If we zoom into the base of the flywing during active flapping, we can see the wing hinge. It consists of radial stop, shown in red, plural wing process, shown in yellow, and Territorial C, a putative mechanisensor and damper, shown in blue. The radial stop contacts plural wing process in 4 different modes, mode 0, 1, 2, and 3, as shown here. In this scanning electron microscope image, we see how the radial stop connects with the plural wing process in 4 different ways from mode 0 to mode 3. Here is a video of the wing engagement at the start of flight as the radial stop moves from mode 0 to higher modes. Notice the shift in the wing amplitude from very low to very high within a single wing stroke.
So watch as the wing engages. So the red part engages with the yellow part right about now, and you can see that it immediately changes the amplitude of the wing.
Engaged, the wing hinge shifts between the different modes and the wing moves at high amplitudes. This is akin to the gear change operation in automobiles.
Okay, so I'll just sort of stop it here. Just to say that there is a gearbox, and this is now an important part of the whole system, and we are now addressing the gearbox hypothesis. We also have hypothesized the clutch. We always sort of figure out how it works. It's again very difficult. But what Tanvi showed through a long series of experiments, and the second part of it just got published last year, is that the wings are coupled oscillators. So wings and the hot-ears are dual coupled oscillators. Each of these is an oscillator in its own right, but they're coupled by a weak linkage. So this linkage here is a weak link. This is a strong link. Okay. So the wings, the two wings are always constrained to move together, but these are not always constrained to move together. So if you look at this graph, up to a point, if you cut the wings, the wing beat frequency increases, and if you keep on increasing it, then up to a point the wing keeps pace, the hot-ear keeps pace with the wings. But beyond that point, hot-ear goes back to its original frequency and the wing continues to rise. Okay, so I have very little time, I think just maybe 5 minutes. I'll just show you a movie to show where we are going next. Oops, yeah. with this. Yeah, so what we are going to see now is all the the muscles that underlie the wing hinge. I told you that this wing hinge was an extraordinarily complex wing hinge. So these are all the skeletal parts. This is the linkage, the strong linkage, and then there's a sclerite at the base of it. Sclerite is just a fold on the insect cuticle, and this is actuated by muscles. So you have two muscles that actuate the first axillary sclerite. Then we have the second sclerite, no muscles here. Then the third sclerite, and this has, as you'll see, several muscles that actuate it. And so I spoke about the concert that goes on for an insect to do exactly what it does with its wings, these fine modulations. This is the challenge. We have something like 18 pairs of muscles which are, configuring this hinge, and we need to understand what is the code that is being used for wings to move precisely the way that they move. So there's a lot of these muscles. I'll just skip. This is sort of a summary of all of that. And so we've been trying to get at this question in various ways. One of the things that we've done is collaborated with my colleague Getty Hassan and a student Sufia, and we looked at how an air puff is translated. So an air puff causes insects to fly. So how is it translated all the way to wing movement? And this can only be done in Drosophila because of the genetic toolbox that they have. So this is almost my final slide. So this is what it comes down to, is we are trying to explain things like this. It's a simple act of an insect landing. And in this case, we have two views. So we have the complete view of the kinematics, both body and wings. And you can see that somewhere here, the wings start to do something different. You can see that in these plots of the wings. And so we now understand that this might be where they just disengage their wing from using the clutch. And where we really need to go with this is to start explaining things like this. So this is a territorial chase. So here's a male fly going after another male fly. This is all happening in 3D at extraordinary speeds. But these are the kind of things that engineers see and drool over, because we simply can't generate things like this. So that's a aggressive maneuver in which the fly is sumo wrestling with this other fly. And then if it finds a female, however, it is a much more gentle creature, so it carries the female. You can see that it's moving its wings through very large angles. Again, we know how. It's going to gently deposit this female, and then as soon as he's done depositing the female, you'll see that the amplitude suddenly goes down because he doesn't need to generate as much force anymore. So I'll stop there. I'm sorry I've gone a little bit over time. These are all the people who worked with me over the years. I have several collaborators on each aspect of the project, many that I've simply not had the time to talk about. But the people who really make it all work are these people. So this is the lab as it. This is the last major outing that we had as a lab, unfortunately, which was before the pandemic. But there's many people to thank, and above all, the Air Force Office of Scientific Research, which is the only kind of organization that seems to look at this kind of research and think that there's something of value in it. So I'll stop there. Thank you.
Condition, they have a movement, either it is right-handed movement or left-handed movement. Do you find similar kind of maybe because of disease or because of wings disorder, insect they do fly like that? And to extension of this like queries, any particular disease is associated with insects?
Yeah, insects have diseases just like we have diseases. You know, most of us who breed insects know this
like
painfully.
any specific disease restricted to specific class of insects?
Pretty much. I mean, take a disease and you'll find that insects have it. I mean, muscular degeneration, for instance, you find in insects. You find tumors. You find all kinds of things.
And what are the right-handed and left-handed movement?
That I don't know. Anything that breaks symmetry is going to cause these kind of problems. But what's remarkable is they are able to deal with it many times. So if you cut their wing a little bit, they can manage.
The question. One sec. There was a question.
Sir, in the dead bug experiment, the body of the bug would be repository, the same body, or like which body you use in that?
Sorry?
In the dead bug experiment, so
Yeah.
which bug body you have used? It is the same one, or you just make it synthetically and sort of?
No, it's not synthetic. It's the actual insect. I'm sorry I called it a bug. Bug is just a slang. It's a fly. But it is, it's the actual, so it's a freshly dead fly. It's the same body, no synthetic.
And how you control that? Through the controlling of the neurons, or like you add into that?
Nothing, we move it with hand. That's how we move the wings. That's what you asked, right?
Okay.
Sorry, in the dead bug experiment,
Yeah.
we're just moving it with hand.
With the manually you're doing that?
Yeah.
Okay. So have you like, what sort of like microcontrollers we can use in order to make it like automatic and sort of stuff? Because we are using the RoboFly sort of word, no?
Yeah, so I mean, those are two different things altogether. I mean, we've tried to actuate it in different ways, but it's not required. I mean, it's a very simple experiment just to demonstrate, the underlying phenomenon, the fact that this is a physical connection, not a neural one. So that's all. It's a simple experiment.
Thank you, sir.
Yeah.
So, maybe a stupid question. You said the nervous system is very sluggish compared to the mechanical oscillators. And you try to explain that. I haven't understood it. Can you repeat in one sentence?
Yeah, so the nervous system, so if you look at so the sort of the language of nervous systems is what we call spikes or action potentials. Now, an action potential takes about 2 to 3 milliseconds. Okay, that's the length of an action potential. We are looking at systems that are going up to 1000 hertz. So clearly, it's happening within the time, sort of time duration of a single action potential. So
you're saying these mechanical oscillators are autonomous and they have their own technique.
Exactly. So they need the nervous system. So it can't work without the nervous system, but they can do many things in a single wing stroke. And this, of course, is a constraint as well as an advantage.
Yeah.
Just so my question is an extension of that, as a matter of fact. You're saying that the nervous system does control flight in some ways, but the flight is autonomous in its own. Also, there is other kinds of sensory mechanisms that initiate flight. For example, light through the eyes and so on. How do those connect to these autonomous movements?
Very good question. So they are much slower. So what they do, vision, for instance, is far more slow than say mechanosensors. So what I'm talking about is really what allows insects to control flight while they're flying. But then there are things that allow them to, so vision is an important part of it. And so what vision does is guide the insect not on a stroke by stroke basis, not flies certainly, but in a few strokes basis. If you suddenly moved their world, they would take a few strokes before they reacted to it.
So it's a mixture of discrete and continuous.
Yes, so that's actually an interesting question, and this is something that really obsesses me because they're using simultaneous sensory information from many modalities, and the brain has the task of integrating this despite the different latencies, and they're still doing a very perfect job of it.
Yeah.
Yeah, very exciting talk. Just wondering, maybe on the similar line, so antenna is one of the sensory organ. So what is the coordination between antenna and halter in the wing? So fly must be sensing something, then only coordinating those movements, no?
Yeah, I'm smiling because that's basically what we work on. One of the things that we found, so I showed you the halteres. Now the halteres are only there in flies. So I only talked about flies today. I didn't talk about moths. Moths don't have halteres. In fact, most insects don't have halteres. Only flies have halteres. So the question is, if that is the vestibular system, what is the vestibular system in all these other insects? And it turns out that antenna, and that's something that we discovered some years ago, that it's the antenna that enables, but that's a whole other lecture. But yeah.
Taken inspiration from this
because you have the last supporter of your project.
Yeah.
But I just wanted to know, because nature has done what we are trying to do.
Yeah.
So much more efficient.
Yeah. In fact, after the first set of results came out with the aerodynamics, there was an absolute explosion of interest in engineers. And through the last 20 years or so, we've seen a large number of groups across the world now, trying to generate little robotic flies based on the kinds of principles that we are sort of putting out. And it's a hit or miss, because quite frankly, one shouldn't blindly copy nature. Engineers, have invented wheels which nature did not have, and so there are extraordinary things, parallel designs that could do the same thing. So it's a sort of mix of what can be taken and what cannot be taken. But certainly they are paying very close attention.